My background was in virology, but I was gravitated towards immunology because I realized that you can't understand a pathogen until you understand the host-pathogen relationship. So although I didn't want to be an immunologist, I kind of had to be to solve the difficult questions. So a lot of this talk will be about immunology. A lot of it's simple, some of it complex, but I'd like to leave everyone with a little bit of extra knowledge. And so lately we've been playing around with synthetic biology to generate hyperstable vaccines. Okay, so I'm Australian. That's where I'm from. That big red dot, it's actually not a big red dot, it's actually land. And it's not a bad place to work. We got a brand new building, brand new kits, machines, you name it. 3 minutes drive to the beach, the Great Barrier Reef. The campus is built in the Daintree rainforest, which I didn't know, but is actually the oldest rainforest on the planet. Older than the Amazon, and expresses higher diversity of plant life than the Amazon. So it's a highly applicable to bioprospecting, which hasn't really been done before. Introduction to the innate and adaptive immune system. Basically, you got two sets. You've got one set of T cells that act against a threat instantly within seconds. They usually just die and release alarm signals. And then you have the adaptive response, which takes about 5 days to kick in. And that's comprised of B cells and T cells. But they have a better memory than you have. I've read papers with B cells and T cells having memories of up to 110 years. So there are some cells in between. I'm not sure if you can see my cursor. But I guess as humans, we like to box things in so we can comprehend them. But the immune system is so much more complicated than we could have imagined. I was reading a paper today where we've now found over 150 different types of T cells. We have no idea what they do. So there's a lot of work to be done. This is just a fancy schematic that a graphic artist designed, but it just describes the difference between B cells and T cells. B cells fire off their receptor like artillery to neutralize pathogens, where T cells in purple here, they need to touch, they need to feel the cell to see what's inside, to see if it's dangerous or not. And they can scan quite a few cells in an hour. So they're basically just, a scanning mechanism for our bodies. I thought I'd give you a crystal structure that our team has derived. It doesn't actually look like this. This is a different we're presenting it. It's basically just a big blob. So on the top here, you've got your alpha beta receptor, which is attached to your T cell. Down the bottom, you've got your alpha helix, which is attached to the presenting cell. And here you're looking at the epitope. This one is from Epstein-Barr virus. It was the first one ever discovered in humans. And you can see here the focus is on this central tyrosine. So there's no hard and fast rules for how TCRs attach. You can attach sideways, conventionally, backwards. We were hoping for rules, but there are no rules at the moment. Sorry. Okay, so what I was doing in my postdoc was using what were called combinatorial peptide libraries. It took me like 3 months to understand what they actually are, but I'll try to explain it quickly. So you have a T cell epitope, say it's 9 amino acids long. What we do is fix position 1 as an alanine, and the rest of the positions are degenerate. And then we basically just go through the whole 20 protogenic amino acids at each position. So a 9 times 20 is 180 subunits. Now I'll show you what that can achieve in the next slides. Here's an example of one. Here's our first 20 tubes, where you can see we've fixed all 20 amino acids at position 1. And we scan them across a clone which recognizes this epitope. And as I said before, the X's are degenerate. So yeah, I forgot to say that the 180 amino acid, the 180 tubes in a 9-mer CPL contain, I think, about 1 trillion peptides, or every variant of a 9-mer that can exist. So we can scan in an unbiased fashion a T cell across every peptide possible. So if you get a T cell and run it across target cells pulsed with these peptides, these are the kind of data you get. So we were first quite surprised because it sort of busted dogma. So for instance, this minimal epitope preferred A at position 1. And you can see this is A at position 1. So the higher the bar, the more the T cell likes it. So you can see there's multiple bars here, C, K, R, even whatever you want. And so, yeah, we were surprised. So we went to the next 20, where we fixed, again, the 20 amino acids. And it preferred L, which is here. But there was actually a preference for Q. This is a little bit difficult because L is an anchor position in MHC, but irrespective, this TCR liked Q. So if you put them all together, this is what you get. So this is the amino acid, the minimal amino acid that the T cell recognizes. And these are all the 10 positions. And you can see there's not just one bar coming up, there's multiple bars coming up. So that gave us the idea of replacing these amino acids, or even using a combination of replacements to build better agonists. Complex slide, but fairly simple. What you really need to look at is we used the CPL library, which is fully quantitative, to randomly derive peptides and test their EC50 against a clone. So all you really need to know, to see here is the black box, which is the natural target of the T cell. So you can see here in panel A, there's a bunch that kick ass. I mean, up to 3-fold increase. Again, every peptide we tried here, increase. Every peptide we tried here, increase. Not much difference, no difference. And just to prove that, you know, there's nothing strange going on, you know, random peptides performed worse than the native ligand. And so after about 300-400 of these, which is an incredible amount of work, and the whole project was about $5.5 to $6 million from the BBSRC in the UK. We collaborated with data scientists and mathematicians in the UK to build a model. But getting back to the CPLs, a simple way of looking at it is kind of like this. So this one is actually a HIV T cell. It likes a 9-mer. So here's its position 1 to position 9, amino acid 1 to 20. So I was taught in university and in textbooks that we had billions of TCRs that were perfectly designed for a target, a potential target, unknown target. Now, if that was the case, we would only see 9 red dots. So the increase in colour means the increase in output of cytokine from the T cell. Now, what you can see here is there is about 30 different amino acids at different positions, which means that theory is busted. TCRs can see different things. Okay, so I was going to spend the rest of the talk on the mathematics. I'm just kidding. I'm not. But this was derived by our collaborators at Warwick University and validated by other collaborators in the UK. And basically what it does is gets the EC50 of all the peptides we've sampled and builds a model on how many peptides a single TCR can recognize. And this is the result. So you don't really have to worry about other stuff. Here's 0, so here's the index. So these are all the fold changes below the index. So these are all the peptides that probably won't function below the index, but these are all the peptides that can function above the index. And the model performed a normalization line, this dot here, where we could get numbers. So we found 500 peptides 1,000-fold more potent than the wild type, 60,000 peptides 100-fold more potent than the wild type, and 1.3 million peptides as or more potent than the wild type peptide. This was published. It's received, I think, 300-400 citations. Yeah, it's very popular in the literature. But what we have to remember is, while this number seems high, 1.3 million peptides covers only 0.0000234% of all possible tenomers. So in this context, the TCR remains highly specific. They're just not monospecific. So doing a lot of structural biology and functional analysis, we thought to ourselves, two molecules actually don't care what the other is made from. They just care about H bonds and salt bridges and affinity. So we thought, why can't we replace a natural ligand with a synthetic ligand? Now, this is advantageous for numerous reasons. First of all, they're resistant to proteases, these non-natural compounds, which are present absolutely everywhere on your skin, intestines, blood, yeah, everywhere. They're quite cheap to synthesize. I did a quick back of the envelope calculation, and I calculated we can make around 22,000 doses of influenza using this technology for one dose that is currently used at the same cost. And it can be upscaled to the megaton. No cells are needed, so it can be completely clean contained. So yeah, advantage to their manufacture. Now, they don't require refrigeration. Every single vaccine requires refrigeration, either on ice or 4 degrees. It can't vary from that temperature more than a few degrees, otherwise it has to get thrown out. And who calculate, or the World Health Organization estimate that around half of all the vaccines we make every year are thrown away because of this chief reason. Now, this helps immensely with populations that are hard to reach, because you don't need refrigerated storage facilities, refrigerated trucks, refrigerated airplanes. I've seen footage of people like going down the river with an ice box. You can keep these things on the shelf for years. And theoretically, they should still be effective. And what I'm hoping to achieve over the next very, very short while, we're doing these experiments now, is I'd love them to be orally active. Because one of the surprises I found was that 20% of the global population have a needle phobia, which means they don't get vaccinated. So if we could turn these things into a tablet, we can get extended coverage. And also, try vaccinating a 2-year-old. It's not very pleasant. I'll just go over this quickly. This is sort of where the synthetic biology comes in. This is what's called a pristine meteorite, called GRA9522. And it gives us a rare insight into extraterrestrial chemistry. It's very small, 128 grams, found in Antarctica, so it was uncontaminated by microbial life on Earth, which is a big deal. It crashed in 1995 or before then, either 1994 or 100 million years before then. They don't know. Anyway, NASA took this thing back to the labs in the US, cut it up, had a look inside, and they found the building blocks of life, all the amino acids. But what they found were these amino acids were found in equal amounts in either D form or L form. So back to front, like your hands. Now, all life on Earth is built with L amino acids, you, your dog, cat, your plants, everything. And it's unknown why. We know deformed proteins are functional, and some people theorize that it was just a flip of a coin, and it was a choice of Earth to choose L amino acids to create life. And there are people that believe there are organisms in the universe that are built back to front with D amino acids. So what we did was, again, with the CPLs, we made a CPL with just D amino acids, and we ran a flu peptide across this. This is, I'm just showing position 1, 2, and 3 here, but what you can see is there's differences between the two. So what I did was built a epitope based on these signals. Is everything okay? Anyway, yeah, completely out of D amino acids. I thought, oh, will this work? So what we did, we chucked in the wild type peptide from donor 1, donor 2, and waited 10 days in the incubator, and then measured how many anti-flu T cells there were in the culture. So in this donor, there was 28%, in this donor, there was 6%. We chucked in GPP, and to our surprise, we found equal numbers of anti-flu T cells. These things are hyper stable. We threw them into 50% human serum, and the dotted line here is the wild type flu peptide, which I was surprised at. It's basically gone in 10 minutes in just 50% serum. And the controls were all okay. So if you can see the synthetic, which is the black box, it's fine after an hour. We thought we'd throw it in simulated stomach acid, human stomach acid, minus all the proteases, which can be up to 250. And same thing happened again. The wild type peptide was gone in 10 minutes, and the synthetic was having a bath after 3 hours. We upped the structures now, but we did modeling, and we found that even though they had different primary sequences, when you put them together, they kind of look the same in 3 dimensions. And that's what T cells look for. We did an in vivo experiment. We did this 3 times with the same results. We got a HHD mouse, which has a human HLA-A2 allele, and a mouse H2D allele and mouse beta-2M. So the T cells can recognize it. We primed them, boosted them, and infected them with either lethal or semi-lethal dose of PR8, and had a look at survival. And 8 of 8 mice vaccinated with synthetic peptide survived. 7 of 8 with wild type peptide survived, and 70 and below with the relevant peptide or adjuvant only. Now, what was pretty cool was we actually fed this stuff to the mice every week, once a week. Not a lot, only 100 micrograms in bicarbonate soda, and bicarbonate sodium. And we had a look at their lymph nodes, and we found flu-specific responses in their gut. Now, this is pretty cool because we didn't use any adjuvant here, and it provides proof of concept that we may be able to vaccinate against viruses using the mucosal tracts, either the oral or nasal. The more peptide, the better. That's basically, this is what this graph is showing. I did the calculations, and it'll be about a gram a day for a human, but we haven't titrated or done any routes of administration testing. This was published in the Journal of Clinical Investigation and got a highlight in Natural Chemical Biology. We're now moving on to synthetic versions where we can increase the pace of manufacture and design. And they are promising. Here's a different peptide on the same allele. This is from Epstein-Barr virus. Here's the wild type, 2%. Here's the synthetic, 5%. Control, nothing. And here is another peptide. This is from a different allele. 9% wild type, 21% synthetic, and nothing for the control. And these are just some examples showing that it's reproducible across donors. So all 4 donors respond. So here's wild type, synthetic 1, synthetic 2, synthetic 3 didn't work. We find that occasionally. I don't really want to spend the money to find out why. And no peptide control, which we didn't get a response for. Here's another peptide that we tried, and this time, this one likes synthetic 2. And we're getting good significance there. And again, the human serum tests, 100% after 2 hours for this peptide. So they're very, very stable. We were worried that they might change the T cell receptor repertoire, leading to God knows what, but it looks like they don't. So each of these colors is a single clone And basically what you can see is red dominates most of the time in wild type and synthetic 1 and synthetic 2. And red dominates in a second donor as well. So it doesn't vary the T cell compartment that we know of so far. Again, we're working on various synthetics. We just want to mine it to get the best, most potent synthetic, and hopefully the cheapest. So here's wild type, here's synthetic version 3. Okay. Okay, this is all right, a synthetic version 5, and no peptide. So I'd just like to leave you with this message. Human T cells can recognize millions of different MHC ligands, but these ligands don't have to be natural. We can create them. We can modify biology. And with that, I'd like to thank everyone involved in the study. And I'm happy to take questions. Thank you.
Thank you for the nice presentation. I just want to know, with this synthetic vaccine, do you still add those preservative agent, which cause most of the allergy?
No,
Hello.
I've actually been keeping it on my roof for about 2-3 years now. To see if it works. I've actually been keeping it on my roof in an Eppendorf tube, just to see how tough it is. But, you know, we've boiled it, left it on the bench, put it in the incubator. You know, as long as you don't go above about 80 degrees or so, they're pretty tough. Most of the problem with peptides is the proteases, because there's thousands and thousands of them.
Okay. Okay, thank you for the response.
Now, I was just thinking that I would be curious if it should be an application of this kind already ongoing for COVID-19 vaccine.
They're cooking right now.
Okay.
The main problem, so COVID's a problem. Like if you look back into the first COVID epidemic in southern China, if you look back at some old papers, they found that antibodies disappeared 2 months after infection, which is what they're seeing now with COVID-2. So, you know, you can't get vaccinated 6 times a year. So I think a combination of antibody and probably T-cell vaccines are required. The problem with T-cell vaccines is Hla diversity. Now I can calculate, or not me, we can calculate probably 95+% coverage across the Caucasian population. It could get pretty high across Asian populations, but for these rare populations, you'd have to generate custom vaccines. Because remember, these things can't be cut. So you can't put them you know, in a line. So actually what I can imagine in the future is, you know, when you're born, you know, get your genome sequence, get your HLA type, and you get your custom vaccine ordered in the mail based on your personal HLA type. That's what I imagine.
Thank you.